Wow. I mean, does that make sense to you? Makes sense to me. I mean, to me, it devastated Behe. Behe's argument was destroyed. Irreducible complexity. I mean, he just showed subsystems that were functional. You know, you could reduce this flagellum and not lose all function. You got this toxin injector syringe that still works, right? I mean, that's a great argument. And also, all the little subparts of this, they work in other systems. Almost all of them, not every one of them. But most of them have other jobs elsewhere in the cell, doing other stuff, right? So that's a really good argument. Let me ask you though, according to Kenneth Miller's argument, which one of these things would you say evolved first? Everybody who votes for the TTS, raise your hand. According to Kenneth Miller's argument, who do you think would evolve first? This one or that one? How many think that the flagellum evolved first? How many people would think Kenneth Miller said the flagellum evolved first? And then the TTS system. No, he believes that the TTS system evolved first because it only requires 10 proteins. This one requires at least, some people would say, he argues 40 or 50, and some people argue downwards as low as 30, but a lot more, right? And how many proteins does he take away to get this one? He takes all except for 10 away, a whole bunch. He doesn't just take one away, he takes 40 or 30 of them away to get this one, and still functional. And he thinks the evolution went that way, uphill. Well, let me show you that TTS system is found in a very few restricted bacteria, gram-negative bacteria that attack plants and animals, which are thought to have come along billions of years after flagellar motility was already here, even according to mainstream thinking. The flagellum, on the other hand, is found in all kinds of bacteria, from pathogenic to non-pathogenic. All kinds of bacteria have flagellum, but TTS systems are very restricted. They're specialized. And there's been fairly recent evidence published in 2008, this paper, where they have found, they actually in real time demonstrated that these parasitic bacteria they degenerate from the flagellum. They start out with a flagellum, and then they've demonstrated all the different little pieces being removed all the way down to the TTS system, and then the TTS system is retained because it's still functional. But it actually has been demonstrated to be a regressive feature, a devolutionary feature, a parasitic feature, if you will, where the TTS system is maintained because it's so much cheaper to maintain. It doesn't cost as many genes to maintain. And so if you're parasitic, you don't have to work as hard to maintain your genome. And so this has actually been demonstrated in real time now. So it's devolution, not evolution. Kenneth Miller is wrong on that aspect. But what about the mechanism of evolution, where he argues that you can get different little pieces and parts that do different little systems in other parts of the cell, and then bring them together to form a more complex, unified structure and function? According to the syllabus, natural selection, describing natural selection, evolution, there are various examples of evolution in action. For example, the evolution of HIV resistance or AIDS resistance, antibiotic resistance, malaria resistance, flightless birds, different finches on different islands, etc. All of these are mentioned in the syllabus here at La Sierra as evidence of evolution in action. For most of these, do you notice a kind of a general theme? These are based either, let me just tell you, antibiotics, most forms of antibiotics resistance. Do you know how most forms of antibiotic resistance evolve? First, you have to know a little bit about how they work. Basic thing on how antibiotics work is they come into the bacterium, they target a specific target within the bacterium, they latch on and disrupt its function. And then the bacterium either gets suppressed or dies because of that. And so what does the antibiotic or a bacterium have to do to become resistant to the antibiotic? All it has to do is make a little single mutation on that target. It makes a little bump, sticks out a finger, and here comes the antibiotic. And can it bind to the target anymore? No. Is the bacterium now resistant? Yes. There's many different ways to make little bumps on targets. And in fact, it's disrupting a previous established interaction. Why is it so much easier to break Humpty Dumpty versus fixing him again? Do you know why? Because there's so many different ways to break him. There's only one way to fix them, right? Same thing with antibiotic resistance. There's so many different ways to get antibiotic resistance. That's why it happens so commonly and so rapidly. And that's why it's such a big problem. It's at the lowest form of what I call evolutionary potential. It's very easy to achieve because there's so many different ways to do it. Same thing with the evolution of HIV resistance. Do you realize that just this year, February, just this month, there's been a research done where they have done bone marrow transplants on HIV/AIDS patients, and they've transplanted T cells into these patients that are missing a specific portion of the CD4 receptor, which HIV virus likes to bind to in order to infect the white blood cell. And given this mutated form of this docking receptor protein, the virus can no longer dock effectively, and these people become immune to HIV and no longer develop AIDS symptoms. They recover from AIDS symptoms. And this has just been published this month. I think that's exciting, but it's also telling on the way that these different forms of resistance work. Same thing with flightless birds. Is it easier to gain a wing or to lose a wing? All you have to do is make one little mutation and you lost your wing. Same thing with blind cavefish. 1 point mutation, cavefish don't grow eyes anymore. Just 1 point mutation. And so there's so many different ways to mutate your eyes or these big complex structures out of existence versus the very complex and very specific ways there are to gain them. A bit about the mechanism. Random mutations. They are real. They happen. There's no problem. There's no argument there. And they must affect DNA in order to be inheritable. And they must affect, depends on the type of reproduction, but in sexually reproducing organisms, they have to affect gametes. Natural selection is also a real force of nature. It's a mindless process, has no goal in mind, it doesn't care about anything. However, it is capable of producing certain phenotypic changes. However, not all genotypic changes produce phenotypic changes or physical changes. The underlying genetics can be changed without resulting in a physical change, and those type of changes are hidden from natural selection. Nature can't see genotypic changes that don't result in physical changes or phenotypic changes. The mechanism of random mutation in natural selection is able to explain very well similarities between different creatures, but it's unable to explain functional differences beyond what I call a very low level or threshold limitation of functional complexity. And I'll get into more on the functional complexity in a little bit, but more about homologies. Homologies or structural sequences or structural similarities are easy to explain by random mutation and natural selection, but the real problems are explained in the functional differences. Lots of things look very similar and even function similarly, but may have key functional differences that have significant structural threshold requirements. The hypothesis of common descent does not trump, in my view, the hypothesis of common design unless one has a mechanism that is capable of producing both the similarities and the differences, the functional differences. The nested hierarchy or the tree of life, the nested hierarchy pattern do not necessarily indicate common descent or common design. A nested hierarchy is kind of like those little Russian dolls where one fits into the other, and they all kind of branch out, and this creature is more closely related to that creature, and then they branch down, and they form this tree of life, this branching pattern. There is kind of a nested hierarchy of the tree of life. And the argument is that this nested hierarchy can be explained by random mutation and natural selection, and I agree. The pattern itself can be explained, but the functional differences cannot necessarily be explained. And also, nested hierarchies are not always produced by non-intelligent processes. For example, humans often use nested hierarchical patterns, the same as this basic tree of life, in deliberately designed systems. For example, in computer systems, object-oriented computer programming uses nested hierarchical patterns in programming. And it's an information system, which is interesting. Also, modern classification systems of living things were originally produced by creationists, like Carl Linnaeus. He's the father of modern taxonomy. He believed that the orderliness of arrangement and the predictable pattern, the hierarchical pattern of living things, was a reflection of the orderliness of the mind of God. And his seminal book, Species Planetarium, he quotes David in the Psalms 104:24, "O Lord, how ample are thy works, how wisely thou hast fashioned them, how full of the earth of thy possessions is of thy possessions." So he believed that this was all a reflection of the mind of God, this whole classification system, the orderliness of living things. So what is the basis for removing the need for God? What's the arguments of those like Richard Dawkins and the like? Well, where the rubber really meets the road, at least in my view, comes to an understanding of DNA. DNA is made up of 4 chemical letters, A, T, C, and G. The information is coded in the sequential order of these chemical letters, just like the Morse code or English or computer codes or any other language system you can think of. It's the sequential order of the characters themselves in the alphabet, whatever type of alphabet you choose that's important. Computers have how many letters? Zeros and ones, right? 2 letters. English has 26, and DNA has 4, but it's still the basic idea, right? The order of the letters code for information, meaningful information. Transcription and translation. First, this information, once you have it in DNA, it has to be turned into a working copy called RNA, or messenger RNA, and then the message is translated into another language system of proteins. How many letters do proteins have? 20, right? You got 20 amino acids, basic building blocks, and so you got to translate from the language that uses 4 letters into the language that uses 20 letters, and that process is called translation. DNA controls what, when, where, and how many proteins are produced ultimately. So decoding DNA. 3 letters in DNA code for a sequence called a codon, and 1 codon translates into a single amino acid letter in a protein chain. There are 20 different amino acid letters in the chain, just like there are 26 letters in our alphabet. And exactly like our English language system, the order of these letters codes for information. And the proteins themselves, their functional shapes depend on their 3-dimensional structure. Proteins are the functional units of the cell. They are very specific in their sequential order. Most of them at least require very specific order and 3-dimensional structure in order to work properly or even at all to do a certain type of function. There is some flexibility that most protein-based systems can realize without a complete loss of function. However, changes in DNA that result in protein sequences can result in either beneficial changes, improved functionality, or decreased functionality, detrimental changes, or neutral changes. Some changes can happen that do not affect functionality. Natural selection. Syllabus quote. According to the syllabus, evolutionary theory must explain how new species originate and how populations evolve. Microevolution is defined as consisting of adaptations that evolve within a population, confined to one gene pool. Macroevolution refers to evolutionary change above the species level. So what really is a species? Before we can go on, we kind of have to consider what they mean by this micro and macroevolution. What is a species? According to the syllabus, the biological species concept emphasizes reproductive isolation. Species is a Latin word meaning kind or appearance, like the Bible uses kind, right? Biologists compare morphology, physiology, biochemistry, DNA sequences when grouping organisms into these species groups. So what about donkeys, horses, and mules? All defined as different species, right? Yet horses and donkeys can get together and produce a viable offspring. Mule. So are horses and donkeys, do they have reproductive isolation? Not, at least in the first generation. What about what about the mule, though? Mule's sterile almost always, right? So is the mule therefore reproductively isolated, and then it fits the definition of a species, right? Let me explain. The reason why a mule is sterile is not because it has unique information that the donkey or horse didn't have, or also because the donkey and horse can mate and produce a viable offspring, means that they have essentially the same quality of information. The problem with the sterility problem, I guess, with the mule is not because of information, it's because of gross chromosomal arrangement. What happens is that there's a chromosomal inversion in the horse relative to the donkey. And so you have this flipped chromosome effect, and when they try to get together during meiosis, they have to make these loop structures in order for the informational sequences to match up properly. And then when they start recombining with each other during meiotic recombination, they start to fragment and they lose information, and you have non-viable gametes. So that's the reason why the mule is sterile. It's not because of an informational difference or an informational difference in the gene pool. It's because of a structural difference in the chromosome. So to me, this is a rather subjective definition of a species, reproductive isolation. Different kinds of dogs, for example, experience extended reproductive isolation, both geographical and morphological, like a Chihuahua versus a Great Dane. It's very hard for a Chihuahua to mate with a Great Dane. Yet all dogs are classified as the same species, right? Why? Because of convention. We all know where all dogs came from the wolf, and so we just classify them as the same species, even though they are, in fact, reproductively isolated, arguably. Compare this to fossil coelacanths, the fish that's supposed to have crawled out on land and started walking, you know. Fossil coelacanths are classified as different species versus those, by the way, they died out 80 million years ago in the fossil record, but they were found alive and well in the mid-1900s off the coast of Madagascar, and where did they go for 80 million years? And they're still alive. Anyway, beyond that, they're classified as different species in the fossil record versus the ones alive today, simply because it's believed that they're reproductively isolated, even though they look no different as far as their structure goes from a Chihuahua and a Great Dane, or a German Shepherd and a Pug dog. So why are they given these different species names? They all have essentially the same kinds of allelic options, I would argue, in their gene pool, despite their reproductive isolation. If pairs can mate and reproduce viable and often fertile offspring, their genetic information is essentially, I would argue, the same kind, qualitatively, of information. Here's a picture of the coelacanth. That's the live one that was caught off Madagascar, and here's a fossil one. And here's the bony structure. One is much larger than the other, 1.8 meters versus 56 centimeters. But despite the size, they both essentially have the same basic bony and soft tissue structure. Yet they're classified not only in different genus groups, or species groups, but also different genus groups as well. What about lions and tigers? You can mate them and produce giant ligers. Look at that huge thing. Polar bears and grizzly bears, they can produce pizzly bears, right? Which are not only viable but fertile. Well, no one knows about the pizzly yet, but these are fertile. What about the horse and the zebra? That's real. That's a hybrid, a Zorse, right? Are they really different gene pools? Really? As far as a qualitative sense? How about a humanzee? Anybody believe the humanzee one? This is humanzee. His name is Oliver, and for a long time, many years, a lot of people suggested that he was actually a hybrid between humans and chimps. And there have actually been unofficial experiments done in the underground literature between mating humans and chimps. But so far, no validated successful results have been produced until Oliver came along, and they thought he was a real hybrid between humans and chimps, and therefore we are closely related. And then was later discovered when they did detailed DNA analysis, that he was just a regular Congo chimp who liked to walk upright. Right? And so this also begs the question that if you can't even tell a missing link when you have a living one, how good are you going to be when you just have like a tooth or a jawbone? Right? What about jackalopes, rabbits, and antelopes? You think they can get together? They make a viable offspring? So if you don't believe this one, what's the difference? Why can't they get together, but the other ones can? There has to be some qualitative difference in the gene pool, which makes these things unable to be produced, right? So differences that aren't. Kind of all starts with Gregor Mendel. He's the father of modern genetics. it's kind of surprising, or at least interesting to me, that almost everything that Darwin published as examples of evolution in action, like his finch beak differences and various differences of different kinds of dogs and whatnot, these are arguably based on Mendelian variation and aren't really Darwinian in nature. They're completely explainable by Mendelian variation, which, just in brief, you get the parent and the mother and the father together. They have their gametes get together and they do these crossovers at very specific places in the DNA. And so the identical qualitative portions of the chromosome on one side gets crossed over with the same thing on the other side. And so you don't get these unusual mixes and matches like jackalopes, right? So, and you can get different expression of phenotypes this way. You can get green plants, or yet green plants with green flowers and yellow flowers, and parents like this, and they can produce offspring that are just yellow or just green, right? All these variations you can do. And for our illustration purposes, let's say I have a bike shop. And at my bike shop, let's say I have the option for smooth tires and knobby tires. And I have the option for these big handlebars and the little narrow racing handlebars. And I go to the bike shop and I can make a bunch of different combinations, right? I can combine racing handlebars with knobby tires, right? And I can buy wide handlebars with smooth tires, or any kind of combination. And at my pool of options, right? But what happens if I put the knobby tires where the handlebars are supposed to go? You're not going to get a very good bike, right? So if you start screwing up with the location of your options, you start destroying the functionality of your creature or your organism. The same thing happens with Darwinian-style evolution. If you want to go and turn a lizard into a bird, can you do that by going to your bike shop and picking one of your options and saying, I would like the bird option? What if you don't have any bird options in your gene pool, right? No feather options yet. How do you get feathers outside of, can you get feathers by Mendelian variation? No, they're not there in your gene pool. So how do you get feathers to evolve onto your lizard? You have to do that by random mutation and natural selection that actually changes the gene pool of options. So what are the odds? In order to do this, in order to create new allelic options for your gene pool, like I don't have any purple tires in my bike shop, I need purple tires. So what are the odds of getting purple tires? That has to be done by random mutations to what you already have, your knobby tires that are gray or yellow or whatever. So these have to do with statistics. What are the odds of getting something new? And I propose to you that the odds are based on what's called sequence space. Sequence space is actually hyperdimensional. There's as many dimensions as the size of your sequence is long. So if you have a sequence of proteins that is 500 amino acids long, you have 500 dimensions. Okay, but since I can't think in 500 dimensions, I like 2 dimensions. Okay, so imagine sequence space as a big 2-dimensional checkerboard, and I start out on the shallow end of the checkerboard where I have short, small little sequences. Well, those short, small sequences will have beneficial sequences that are closer together, represented by the green dots. So it'll be easy for me to randomly step from one green dot to the other green dot without hitting a square that has no green dot, right? But as I go higher and higher levels of complexity, longer and longer sequences, the green dots get rarer and rarer in an exponential manner. So me taking a random step and hitting on a green dot of a certain size of my step, the odds become exponentially less and less. Same thing, it's a stepping stone problem. How long before you have to swim for it, basically? So I'll illustrate this with English language systems. For example, 2-letter checkerboard size for English language system words, there are 677 options, or 26 to the power of 2. 3-letter checkerboard size, the number of options is 17,576. So there's a huge increase in options, but there's also an increase in defined 3-letter versus 2-letter words in the dictionary, right? But look at the ratio. You go from a ratio of 1 in 7 to a ratio of 1 in 18 of defined versus not defined. But it's still pretty easy to evolve at low levels, right? I can get from cat to hat to bad all the way to dog by taking one little character changes at a time without destroying meaning, right? So you can get from cat to dog quite easily without destroying meaning just by taking a random walk along connected stepping stones. However, what happens is this random walk so easy at larger or higher levels of functional complexity. Look at the ratio, what happens up to 7-letter words. The ratio goes from 1 in 7 to 1 in 18 to 1 in 350,000. And this is not just words, this is 7-letter sequences, combinations of like 5 and 2-letter words, and if you want to throw spaces in there, it gets even better. And we're not even talking beneficial here. We're just talking meaningful versus not meaningful in the English language system. And see, you can kind of visualize this as an expansion of the water versus the land in your stepping stones. And so as you go on higher and higher levels, the stepping stones, they kind of get stretched out and pulled apart like chewing gum. They get these narrow little bridges, and pretty soon at higher and higher levels, even the narrow little strands of the bridges start to break apart, and the islands in sequence space become truly isolated from each other, the beneficial islands. And so what happens to the random walk? How many random blind steps do I have to take before I land on something that is actually beneficially selectable by natural selection? Well, the little red line represents the random walk, and the yellow line represents the Hamming distance, or the absolute number of character differences needed to get from A to B. So as you increase the Hamming distance, the random walk distance increases exponentially. And here's a three-dimensional view of random walk. The hyperdimensional aspect of the problem only enhances the problem. Even higher levels of powers of ratios. So how long would it take in English language example? Let's try a 28-character sequence. Changing 1 letter per second with no repeats, it would take over 3 million years to come across just one of 100 trillion meaningful phrases on our checkerboard. And there's the math. So how about increasing the population size? That's only, the previous calculation is only using one individual randomly walking in sequence space. So what if you use like trillions of them? Take the same scenario as before, 28-character sequence space, and increase the number of evolving phrases or individuals in the population by 1 trillion. Now you have 1 trillion random walkers walking around searching for beneficial sequences in sequence space. This would reduce the search time by 1 trillion-fold. It would go 1 trillion times faster to find your beneficial targets. So it helps to increase the population size. The problem is that local environments are limited. They can only tolerate a certain size of population. So your population is limited. And so what happens to higher and higher levels of sequence space or of functional complexity? If your population is limited, how long does it take for your levels of complexity to outpace your population growth? For example, imagine a 1,000 character sequence space, and you have 1 trillion individuals in your population. How long would it take any one of those individuals in that population to find any one of the beneficial characters in trillion-letter sequence space? Or, sorry, 1,000-letter sequence space. It would take 1 to the exponent of 600 years. So, not realistic time frame. So Richard Dawkins to the rescue. He knows about this problem. He argues that all you have to do, and he presented this little scenario. He took a computer. He started with a random sequence, right? And he took the sequence and he made a bunch of copies of it, offspring. And then he mutated each of the offspring. And then he picked the closest match from the offspring population to a target sequence, like me thinks it's like a weasel. And then of the closest match of that generation, he had a bunch of offspring from that one, and he picks the closest match again to me thinks it's like a weasel. And in only 40 generations, he evolved from blah, blah, blah to me thinks it's like a weasel from Shakespeare. Easy, no problem, right? So has he really solved the problem? So question, which is more meaningful? Queasly-Legook or Queasly-Guck in the English language system? They're both equally meaningless, right? So which one is nature, and if nature has to select based on meaning or function, which one is nature going to preferentially select? Neither one, right? It will just toss a coin, I don't know, and it'll select one or the other, but at random. There will be no preferential selection. So going from blah, blah, to blah, 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 is that any sort of thing that nature can recognize? So Dawkins has completely removed the whole half of the equation. He's taken natural selection out of the equation and based it on target sequence comparison, which does work in certain situations, but you can't make a bacterial flagellum using target sequence comparison if you don't already have the target, right? So comparison to real life. This is fine with the English language system. But are there real examples of evolution in action? A lot of creationists will tell you no, but they're mistaken. There are examples of evolution in action. Some examples, for example, there's a nyelinase enzyme. Nylon was not invented until 1935, and then in this Japanese company that makes nylon, they started discarding their old nylon in this little pond behind the factory, and then somebody noticed there's bacterial mats growing on this pond, and they were eating the nylon and getting energy from it, and they say, how in the world did these bacteria evolve the ability to eat nylon? And what they did is they mutated a pre-existing sequence in their genome, and they turned it into a nyelinase enzyme, which they didn't have before. And so that's one example of evolution in action. Another example is lactase enzyme. This scientist, Barry Hall, he took bacteria, E. coli bacteria, that had the ability to digest lactose sugar and convert it into energy. And he deleted the genes, the lactase genes that gave the bacteria the ability to do this. And then he grew them on lactose-enriched media to see if they would ever evolve back this enzyme again. And in a very short time, 1 or 2 generations, they did evolve back the lactase enzyme again using a single point mutation and gained back the lactase using a completely different sequence that did not have the lactase ability before. And so you have to ask yourself, what are the odds? Other examples we already talked about, antibiotic resistance. Sickle cell anemia is essentially the same idea. Flightless birds, blind-caged fish, those are all different forms of evolution which result in a loss of a pre-established function or interaction. So limited evolutionary potential. Most stories about lactase or nyelinase, they stop with the fact that evolution works at that level, but they don't go beyond and tell you the rest of the story. For example, Barry Hall, what he did next is very interesting. He deleted the newly evolved gene in the bacteria, and then he regrew the double mutant colony or bacteria on the same lactose-enriched media to see if they would evolve another enzyme. And they didn't. He watched them for about 40,000 generations, and they never evolved a new enzyme. And so he says, these double mutant bacteria have limited evolutionary potential. So I ask you, what is it that limited the evolutionary potential of the double mutant bacteria? And I propose to you that they were right on the border, right on the margin of the edge of evolution, of where random mutation, natural selection can work in observable time. You go a little bit beyond that, you don't get evolution at all. For example, you can go to scientific literature, look anywhere you want, you will not find an example of evolution in action of producing a system that requires more than 1,000 fairly specified amino acid residues working together at the same time. To give you an idea of what this means, the flagellum requires about 10,000 working together at the same time. ATPases require about 3,000 working together at the same time. No such system or even subsystem has ever been shown to evolve in real time in literature, period. Lactase, on the other hand, it's a single protein enzyme. It requires no more than about 300 amino acids working together at the same time. Nyelinase is about 250 working together. So you can get right up to that level, but anything beyond, there are no examples of evolution in action in literature. And I propose to you the reason for this is because as you go up the level of functional complexity or increased minimum size requirements, because some systems require greater numbers of building blocks than other systems require, as you increase the number of building block requirements at minimum before you gain a certain functionality, you increase the distance between stepping stones. This is a paper published in a journal not too long ago, let's say 2006, where they map on a three-dimensional sequence space proteins at different sizes, from 30 amino acids to 300. And you notice that the smaller proteins are more clustered together, and as you go up to the larger proteins, they are wider and wider apart. Now remember, this is a three-dimensional projection of up to 370 dimensions of sequence space. For example, let's say I have a bunch of balloons hanging in this room in different places, and I shine a light from back there this direction, and I have shadows on the wall. Will the shadows look closer together in 2 dimensions than the balloons really are in 3 dimensions? Yes, they will. And so when you have 370 dimensions projected onto 3 dimensions, how far apart are these things in sequence space? They're a lot farther apart than they look in 3 dimensions. And statistically, they get exponentially and exponentially farther and farther apart. Also, the overall trend is downhill for evolution. Nuclear DNA mutation rates, at least for higher level organisms, the mutation rate for humans, for example, is about 200 per generation. On average, of those 200, 3 will be detrimental, and 197 will be neutral with respect to function. And none will be beneficial, because the odds of a beneficial versus a detrimental is about 1,000 to 1, favoring detrimental over beneficial. If harmful mutations enter the gene pool faster than they can be removed, obviously eventual extinction will be inevitable. So to avoid eventual extinction, every woman would have to give birth to about 40 offspring per generation. So, how many ladies have done their part? Right? So, and this has been published by Crow and others, and they write the reduction in fitness or due to deleterious mutations. They say each female will have to give birth to 40 offspring for 2 to survive without having detrimental mutations in themselves, and so the gene pool wouldn't decay over time. So according to standard population genetics theory, the figure of 3 harmful mutations per person per generation implies that 3 people would have to die prematurely in each generation, or fail to reproduce for each person to reproduce in order to eliminate the now absent deleterious mutations. So humans don't reproduce that fast, and neither do elephants or monkeys or anything like that. So how do we get rid of all these deleterious mutations? Their calculations are interesting. They base it only on 2% of the genome being functional. Well, now recent information suggests a much higher percentage of the genome is functional. Others have estimated that the detrimental mutation rate is actually probably greater than 5 per generation, which implies that the offspring rate would have to be 148 per woman instead of 40. And if you consider that some estimates of the genome, given the functionality of non-coding regions of DNA that are being discovered in junk DNA, suggest over 50% of the genome is somewhat functional to at least some degree, the mutation rate, detrimental mutation rate, would likely be over 30, which suggests a needed reproduction rate of over 10 trillion per woman. So it seems fairly conclusive that we're headed toward extinction, and so what is the argument to explain this? Well, most of the argument is positive, what's called positive epistasis. It's where detrimental mutations are clustered together in a few unfortunate individuals who happen to die and get rid of all the bad mutations so the rest of us can survive. So how do you concentrate all the bad mutations in poor, unfortunate, small groups of individuals? Well, sexual reproduction is thought to be one mechanism to do this, but sexual reproduction only helps if you do the odds on it, the statistics on it. If you look at random processes, it only helps by about 50%. It reduces it by half. So instead of giving birth to 40 children, you only have to do 20, right? Or 10 trillion, you only have to do 5 trillion, right? So it still seems like a huge problem to me that hasn't been adequately explained in scientific literature as far as I've been able to tell. And then people say, well, when are we going to go extinct? Well, I don't know. All this tells us is direction. We're headed toward extinction. And men will go extinct first. The Y chromosome does not undergo sexual recombination. It undergoes only mitotic division. And so the Y chromosome is known in literature. It's going at the full rate toward extinction, whereas the rest of our chromosomes, they got that reduced rate, the half rate for sale price, right? And so us men are going to lose our Y chromosome here shortly. So the question to me is, well, the argument is, well, that's no big deal because other chromosomes will take up the function of the Y chromosome, so it's really not a big deal. So my question is, though, well, how did the Y chromosome evolve to begin with, if it's just kind of this baggage thing? Dating methods. The problem My biggest problem with dating methods is calibration. There's a theoretical range, for example, for carbon-14 dating up to about 70,000 years or so, due to its fairly short half-life of around 5,700 years. Radiocarbon dating seems to be pretty logical on the otherwise, but it must be calibrated because there's not a direct comparison between the carbon-14 date and the actual age. So it's calibrated against other methods. One of the most prominent calibration methods is against tree ring dating. The oldest living tree is the Methuselah tree, a bristlecone pine tree, which has around 4,700 rings in it, so obviously around 4,700 years old. So how do you get older calibration sequences? Well, you can't go far beyond 4,700 years, calibration out to several tens of thousands of years. Well, they do this by overlapping living wood with dead wood, dead trees, and they take the dead sequences and they overlap the rings, and they build them up like this. So here's, let's say this is the living tree, and they found a dead tree, and the sequences match here, and then this other dead tree and the sequences match here, and you get these matching wiggle curves, these wiggle patterns. And so then you can extend your sequence on beyond 4,700 years, right? So there's a bit of a problem, though, with these matching sequences. In 1986, Yamaguchi published a paper where he took 290 ring Douglas fir log of a known age by historical methods, and he cross-matched it to 113 different places in the Pacific Northwest Douglas fir master growth sequence with greater than 99.9% accuracy. And 6 of these matches were non-overlapping. So how do you get 9? I mean, obviously, some of these were wrong, right? And yet they were all very accurate. And so how do you decide which is the right match if all your sequences seem to match with the same or very high statistical value, degree of confidence? Here's another sequence. You got a top wiggle match here, a tree wing match, and then you got another sequence wiggle match here. These two are supposed to match with each other. This is a random sequence, a random generated wiggle line here, right? Which one is the better match? Can you tell just by looking at it or computer analysis? Douglas Kuniholm, he suggests that this is a bunch of voodoo going on here with some of these problems with matching. He writes, in almost all branches of science other than tree ring studies, there's a check on the validity of published research. Other researchers can and often will independently seek to replicate the research. Tree ring studies do not have this check. The result is a system in which investigators can claim any plausible results, and yet they're accountable to no one. So tree ring studies seem to be a bit subjective, to say the very least. The same or a similar problem is found in ocean core dating. You got the same wiggle matches, and then they add this, you see this word, it says tuned here. So they take this original wiggle, then they tune it so that it will match a known sequence. They kind of fudge it a bit and stretch it out and go up and down, and that's called tuning, so they can get it to, because it doesn't exactly match, and so they have to manipulate it a bit. So here's a quote regarding this tuning process and Milankovitch cycles, which are the other pattern that's thought to match. These cycles are thought to match solar orbital patterns and sequences of distances from the sun and whatnot. So at this meeting, they went and discussed this tuning procedure. Mueller scored the most points at the meeting when he attacked the standard technique called tuning that oceanographers use for dating layers and sediment cores. The task of dating these strata is difficult because sediments may accumulate more quickly during some areas and more slowly in others. To tell the age of layers between known benchmarks, researchers often use Milankovitch orbital cycles to tune the sedimentary record. They assumed that ice volume would vary with the orbital cycles. The same thing with ice cores. Then they line up the wiggles in the sedimentary records with the ups and downs in the astronomical records. So you pick the wiggle that matches the best and you kind of stretch it out so it matches. Tuning continued. This whole tuning procedure, which is used extensively, has elements of circular reasoning in it, says Mueller. He argues that tuning can artificially make the sedimentary record support the Milankovitch theory, and vice versa. Mueller's critics or criticisms hit home with many of the researchers. He scared the Jesus out of them, and they deserved it, says Broker. Similar dating problems. Calibration is a big issue for all, or at least what's appear to be the most, if not all, radiometric dating methods. The other argument is that multiple dating methods agree. And to me, it doesn't match much if they're all calibrated against each other. They're kind of, if you know the pattern to begin with, what are the odds that you're going to kind of be able to fudge another pattern to match it? For example, what are the odds that I'm going to be able to find a specific 3 or 4 letter sequence in the works of Shakespeare? Or what are the odds that eventually, if I'm looking hard enough, I'll be able to find an elephant in the clouds outside? Right? Anybody see the toast where the Virgin Mary was found in a piece of toast all burnt into the toast and sold for like $30,000 on eBay? Eventually, if you're looking for a pattern, you already know what your pattern is that you're looking for, you're going to find it in certain types of sequences. But, you know, this whole self-fulfilling prophecy for calibration against each other. Also, this calibration or known methods that agree have been discounted in various situations in mainstream science. For example, Lord Kelvin once estimated the ages of both the Sun and the Earth based on cooling rates at only 25 million years. Kelvin's estimates of 25 million years were supported by 2 completely independent dating methods, gravitational contraction and cooling rates. And yet, nobody believes those ages now. And yet, these are 2 independent dating methods that agreed with each other, and everybody thinks they're both wrong. Other dating methods that don't agree that are somewhat problematic. Erosion rates. Only 10 million years are needed to erode everything on the continents down to sea level, all the sedimentary layers, even from the mountains. The coastlines are also eroding at tens or hundreds of centimeters per year, and yet the continents still feel like a puzzle, and we have sedimentary rock on top of the continents. How is it still there? Why didn't it get washed into the ocean by now? Also, why do the continents still fit like a puzzle if there's such a huge erosion rate on the coasts? Only 2 centimeters per year over 200 million years, that leave you a little dot in the middle, right? Or a distorted coast at the very least. And yet the continents seem to fit together like a puzzle, even though Pangea supposedly was 200 million years ago. Tektites are also interesting. They're these, when an asteroid hits the Earth, it throws up rock, and then these fly so fast that they heat up and they turn into these melted structures called tektites. And these things can be dated using potassium-argon dating and fission track methods. And in Australia, at least, they've been dated up to 700,000 years old using potassium, argon, and fission track methods. However, the organic sediments in these same layers have been dated by carbon-14 at 7,300 years. So there seems to be an interesting discrepancy. The usual argument is that these tektites must have been transported from older sediments to younger sediments. But some of these tektites have very fragile, delicate features on them that haven't been broken or disturbed or rounded off or anything that you would expect by a transport process. So some mainstream scientists have argued that this is very clear evidence that these tektites are in situ, that they weren't transported. And so how do you explain the discrepancy? That's very interesting. The same thing with Grand Canyon lava dams. It was once believed that the lava dams in the Grand Canyon eroded over hundreds of thousands of years each. And then in 2002, a group in Arizona published a paper showing that the lava dams actually, when they hit the water at the bottom of the dam turned to ice, or not ice, but turned to glass. And glass is very fragile, and when there's local tectonic movement, the glass shattered, and all the water that had built up behind it in lakes rushed through the fractured glass base of the dam, and the dam collapsed within minutes and released a huge wall of water that carved away at least the western portion of the Grand Canyon extremely rapidly, like with 30 times the normal flow of the Mississippi River. And so because of the huge flow that was released, they have these exponential decay curves on the side of the Grand Canyon. They reduced the age of the western portion of the Grand Canyon, the Marble Canyon and Inner Gorge, from around 3 to 5 million years old to down to 700,000 years old. So, And the earlier estimates of 3 to 5 million years old, they were based on radiometric dating methods. Same thing happened with Mather Gorge and Holtward Gorge. They used to be dated over 1 million years old, the Potomac River. And then in 2004, beryllium dating was done on them and reduced the age of both Mather Gorge and Holtward Gorge to around 13,000 years old. And a quote here, the erosion rates are tens to hundreds of times faster than scientists had previously suspected. So it seems like there's something that doesn't jive here. Same thing with smooth Grand Canyon domes. Here we've got the Grand Staircase over here, and we've got the Grand Canyon here, and you've got smooth erosion over the surface, and you've got stepwise erosion like this, and you've got these very sharply defined lines that aren't rounded off. They're sharply defined, and these sediments used to cover the whole thing over here, and they got washed away very evenly, which is very interesting to me. For example, here's a picture of a little river here, and here's the sharply defined edge, and you got the layers here, and you got a very smooth surface. And then you got this mini Grand Staircase, right? So how was this thing developed? First there was a sheet of water, I proposed, that flattened out this whole thing, and then the water got less and more narrow, and it got down to here and made this flat, and then this flat as it got narrower and shallower, or lower and lower. And so it seems to me like a similar appearance to the Grand Staircase here, that this was a sheet of water, went fast, carved everything out very flatly and rapidly and very sharply defined, and as it got lower and lower, it receded until all that was left is the Colorado River. So there's a little mini Grand Staircase, and see how flat the surface of it's just sheet-like erosion. How do you make such flatness over slow, long, extended periods of time of erosion? Also, this little butte is interesting. Notice that everything else got eroded away, but the butte survived. The little tiny butte of layers survived. How did it avoid getting eroded with everything else? It did not rain there or snow there or whatever. It seems like it's more consistent with a rapid erosion process where the water disappeared before it was able to take that butte with it. And here's a picture of the butte. See how it's surrounded by nothing for miles and miles around, and it survives. Same thing with Monument Valley. Here are these monuments. These are horizontal layers. This layer matches that layer, matches that layer, and that layer. They were all once connected. How did these monuments survive when everything, all the dirt around them, got eroded away? Is that a fast erosion process or a slow erosion process? To me, it doesn't make sense that these things could survive over millions of years of slow erosion. Clastic dikes are interesting. These clastic dikes are made up of layers that squirted up through higher layers. And you've got, here's this layer, Jurassic, and then Triassic here. And you would think that each of these layers, if they represent millions and millions of years, that they would turn solid or to rock or some form of solid material before the next layer came along. And so my question is, how do you get solid material to squirt, like toothpaste? And, and then this gets eroded away, and then you see these clastic dikes. And it makes more sense to me, at least suggestive anyway, that these layers were laid down rapidly, and while they were still soft and muddy, the pressure from the overlying layers put such extreme pressure on the underlying layers that the sediment squirted up like toothpaste. Found a little defect in the overlying layers, and it squirted before they had a chance to turn to stone. Another argument is, well, how do you get termite nests in the fossil record if they're really rapid deposition like I've just proposed? Here's these termite nests here, and see, you can see all the little tubes and stuff where the termites made their little passageways. Of course, these are kind of random looking tubes. The termite nest that I looked at looked more linear or orderly. This looks like, to me, if you go to the beach and you take wet sand and you dribble it around, and you make these kind of dribbly things. But there's other pictures like this where they're straighter. And so these are proposed to be insect or tubes produced by termites. But Ariel Roth has done some good research recently that demonstrated that these are not likely produced by termites at all, that they're most likely inorganic features. And even the surrounding rock has very similar features and patterns to these tube-like elements that this is the result of spontaneous crystallization perhaps in a hypersaturated environment. And you can talk to Ariel Walth afterwards if you're interested in that particular finding. Kinmer 1470, this is interesting because the layers that this fossil was found in, they were dated before the fossil was found at 220 million years old by 3 different labs using radiometric dating methods. Then the fossil bones were found, and they said, wait a minute, this hominid fossil can't be 220 million years old. I mean, come on, that's ridiculous. So they went back and redated the layers. Until they redated it several times, many, many times, until finally they got it down to 3 million years. And so, is that kind of subjective? I mean, wait a minute. When you know the right answer, then it's easy to date stuff, right? What about ancient ice in Greenland? These ice layers line up, and there's hundreds of thousands of them, which means that every layer is a year, and there's hundreds of thousands of years. One of the really interesting questions to me is that there's this hypsothermal or warm period that supposedly lasted from about 5,000 years and ended about 4,000 years ago, from about 9,000 to 4,000 years. And it was about 1 to 2 degrees warmer Celsius than today. And Greenland, if nobody has checked, is melting very rapidly today at around 200 cubic kilometers per year. And so my question is, if Greenland is melting so fast today, how did the ice survive during the time of the pharaohs for 5,000 years when it was even warmer than today, arguably warmer? And it seems maybe there's a potential problem there. Young river deltas, we can get into that, kind of going over a bunch of different things. Modern river deltas, at least, there's arguments that there's old river deltas that are older. But all modern river deltas are between 4,000 and 8,000 years old, which is interesting. Young ocean sediments. About 30 billion tons of sediment are deposited in the oceans every year. And only about 2.5 billion of that is arguably removed by subduction of plate tectonics underneath the ocean plates every year. And so the ocean floor inside sediments currently total about 1 times 10 to the 17 tons. And that tonnage would be able to be produced by only 15 million years of deposition into the ocean. And the oceans are supposed to be 3 billion years old. If they were 3 billion years at 30 billion tons per year, that's enough sediment to fill the oceans many times over. The ocean can contain 1 billion metric tons of water. And so that's plenty enough sediment to completely fill the oceans over and over. So how in the world did this sediment get removed, get reprocessed, if the oceans are really that old? Fossils with carbon-14. A lot of fossils are not completely replaced by inorganic materials, and they can be carbon-14 dated, and they're showing to be less than infinite ages. And there's, of course, arguments that they're contaminants, but some of these arguments for contaminants or in situ uranium that produces the carbon-14, it's very difficult to support those arguments. And some of the carbon-14 ages are even younger than 40,000 years that are pretty reliably documented, sometimes as young as 20,000 years. And so how did the carbon-14 get there if these fossil carbons are really old, millions of years old?